Doc. Hello, everyone. My name is Sina Ando, and I'm your moderator for this month's session of the African Seminar Series. Our title for today is Improving Health Using Simple Optical Instrumentation and Research Through Training and Capacity Building. And our keynote speaker is Professor Jeremy Zoe, who is a full professor of photonics at the National Polytechnic Institute of Yamoussoukro and the director of the Interdisciplinary Unit of Research and Innovation in Applied Electronics and Electricity. He also leads the Laboratory of Instrumentation, Image and Spectroscopy that he founded in 2007. He has graduated more than 30 masters and PhD and 10 PhD students. He is also the African Spectral Imaging Network founder member and coordinator, and in this capacity has participated to the training of hundreds of young African scientists in the areas of lasers, spectroscopy, photonic and remote sensing spectroscopy. Prof. Zoe is co-founder of the African Network for Solar Energy, and so the board advisor and national representative. He was an associate member of the International Center for Theoretical Physics for more than 15 years and the Third World Academy of Science grantee for 3 times. He is currently a board member of the African Higher Education Board. He is also the representative of Bill and Melinda Gates Medicine for All initiative in West Africa and co-partner at Virginia Commonwealth University, where he has served as visiting professor from May 2016 to August 2017. He has inspired the NIH USA initiative on harnessing data science for health discovery and innovation in Africa, which is currently being put into place. At this point, I would like to hand over to Professor Zoe. We are excited to hear your presentation.
Thank you very much, dear Chair. Do you hear me?
Yes.
Okay.
Yes, we can hear you.
Thank you very much for the introduction. It's a great honor and privilege for me to share with you part of our research in my laboratory. I'm, as you said, Jeremy Zoe. We pronounce it Zoe. I am from the western part of Ivory Coast. My background, my research background is laser engineering. I did my PhD in Paris from 1991 to 1996. And during this time, I was working on two types of topics. I started in nonlinear optics area. At this time, we were trying to generate some new wavelengths for lasers. So we introduced the idea of using organic material as nonlinear material to generate a second, third, and fourth harmonic of some powerful laser. And after this segment of my research activity, I went to another department and I started working on producing metal clusters and looking the magnet optical properties. But when I decided to come back home, I have to decide if I still working in this fundamental aspect of research or working on something more dedicated to, let's say, addressing developing issue in Africa. And the second choice was what I made. And in this direction, I was supported by the international Swedish program of Uppsala University, the Third World Academy of Science, my home institute by providing the space where I implemented my lab, and the research was carried out with a scientific partnership. The main one is from University of Lund in Sweden, and some institute in Ivory Coast. CEDRES is a medical institute working in malaria area. It's also the same for CSRS and Institut Pasteur in Abidjan. Okay. So during my talk, I will introduce our journey towards AFSIN. AFSIN is African Spectral Imaging Network, and then give an overview of light. I have called it an ambassador from nanoscopic scale to remote object. And I will show our research on in the effort of proposing a fast and automated malaria detection instrument. And some work we did in the prevention aspect of malaria. And I will conclude with some instrument we built also for health. And introduce the possibility of partnership with other institutions. So we were 6 countries, Burkina Faso, Mali, Ghana, Ivory Coast, Senegal, Kenya, Sri Lanka, Sweden. We met in Cape Coast in 2009 for a workshop. And during this workshop, a friend and I, we initiated a multispectral and multimodal optical microscope with application to malaria diagnostic. And during this workshop, we made 6 copies of the microscope, which were distributed to the participating countries. And the participants were trained in the area of statistical multivariate methods applied to multispectral imaging. And the project has attracted broad interest. And later in 2010, we were participating to a LAM conference. LAM is a laser atom molecule. It was another network in Dakar. And during this meeting, we organized a session to create AFSIN. The main idea was to propose a space for physicists working in the area of instrumentation to give them an opportunity to develop instruments for their labs. The overall goal of this network is to contribute to the benefit of the laboratories in the members countries to develop international standard research activities in the field of applied spectroscopy and spectral imaging. And our main strategy is to organize an annual workshop for the training of students and young scientists. And the area we target is optics, photonics, imaging techniques applied to medicine, agriculture, and environment. We built instruments for realistic diagnostic tools related to tropical disease, such as malaria, but also we are working in pollution monitoring and natural disaster affecting agriculture, environment, and so on. A significant effort were devoted to training programs. So We are currently a program where we are training PhD doctorates. We started 3 years ago, and we are PhD students from Cameroon, Togo, Kenya, Burkina, Mali, and so on. And we provide a financial support for both scholarship, but also instrumentation. So the basic idea of the instrumentation we build is that as a light source interact with a material, it's possible to take advantage from the intensity of the light, the position, the propagation, the phase, the temporal modulation, the polarization, and the spectral content. And the events, as you know, which occur during this interaction are many. We can just note specular reflection, absorption, transmission, fluorescence, and scattering. And these modifications are used to interrogate the target and for diagnosis, for quality assessment, and many other applications in all the area, in chemistry, in biology, in medicine, environment, agriculture, and so on. So this is the area we address during our research. We cover the micro scale to the macro and the remote study. And the application, as I said, we are interested in medicine, in organic products, and in ecology. And I will try to show you what we have done so far. As you can see on the slide, one of the main activities or the main application of our research is developing instruments for diagnosis in medical area. We use also the same technique to assess organic product. And we also have some study on ecology. And I will develop this aspect later. Okay. Let's now come to this microscope, which was our first instrument. This microscope is a standard type of microscope you can find on the market. It is an Epi Fluorescence Microscope with 3 modalities. We have the transmission mode, the reflection mode, and the scattering mode. And what we did is just remove the white light and replace this white light with a set of LEDs covering the range from UV to near infrared. The idea behind this technique is that when you interrogate a region of interest, you can use a white light. In the image you collect, you have the superposition of all information, all the optical properties. And what we wanted to do is to take advantage of the specific response of each channel moving from UV to near infrared. And one of the applications we have used was malaria diagnosis. As you know, in Africa, when you have a suspicion of malaria, you go to the hospital, they will collect a blood and spread the blood on a slide. And then the slide will be stained with a chemical which will help to bring contrast between healthy red blood cells and infected red blood cells. But all this process is manual and is time consuming. So what you wanted to suggest is a fast and automated instrument. So when we introduce this technology, we were able to make a diagnosis within 3 minutes instead of half a day in the past. So the main things behind this technique is that we replaced the chemical staining with a spectral staining. By doing this, we were able to use image processing techniques, multivariate statistical multivariate chemometrics techniques on the recorded image. As you can see on the screen, each pixel will lead to a vector, will lead to a specific curve, which will be the footprint of the pixel. And if you go to the scale of the cell, each cell will have a very specific footprint, which will be used in an image processing technique to differentiate, to classify, and to give a diagnosis and say if the patient is sick or not. And this gave, this led to a very nice paper in the Journal of Biomedical Optics in 2013. So on this slide, you can have what we typically get in the transmission mode. The image on your left is a combination of 3 channels to bring the color aspect of what we are used when we use a color camera. But the raw images are monochromatic. They are individual wavelength image. But for to give a better perception, visual perception of the image, we made a combination of 3 channels, one in 405, the second in 560, and the last one in 730. So we combine the 3 to give what you have on the screen. So the first one is the transmission mode. The second one is the scattering mode. And what is good with the scattering mode is that you are able to detect the homogeneity in the structure of the cells. And we also have the reflectance mode. So the 3 modes are combined in our algorithm to make the diagnosis. On this slide, you have the main process we use to identify the healthy red blood cells from the infected one. We use an hierarchical classification analysis, as you can see on the screen, with a different step. And gradually, we are able to see if the cell is healthy or not. On this one, as you can see, when the the blood cells is healthy, as you know, the erythrocyte has a biconcave. Sorry. As you know, the red blood cells has a circular symmetry and a biconcave shape. So when the cell is healthy, this symmetry is preserved, as you can see on this HCA classification on this on the screen. So the spectral fingerprint are extracted level by level. And on this screen, you can see 3 different categories using the previous techniques I have shown. In between, you have unhealthy erythrocytes. Below, you have an infected patient blood smear. And this is the later stage. And here you have the very earlier infection. So we demonstrate in this paper that we were able to detect the presence of malaria within the very first hours of infection. That was one of the big contribution of these activities. And later, we developed a number of spectral image processing techniques to target different challenge we faced during this work. The first image is the raw image we collect from the microscope. But from this image, you need to remove the background. So we developed different type of techniques to make it. And then we segment it and then classify. We made a classification and counting of the cells. And then we were able to distinguish between the healthy one and the infected one. And recently, we decided to move from LEDs to laser microscope. The main idea was that using the LEDs, as you know, the spectral, we are a broad spectral band, and it was very difficult to build a contrast function using just 2 or 3 wavelengths. So we decided to use a laser because the laser gives us a very small spectral band. And the second thing we did with this microscope was that we automatize all the process. The sample we were able to move automatically the sample in the X, Y, and Z direction to look at a different region of interest automatically. And another advantage of this microscope was that it is versatile. It's possible to design, modify it, and try different type of modalities. So we implemented a fluorescence microscopy, an excitation microscopy, an emission microscopy, a polarization microscopy, and so on. So we use this microscope to build a new modalities with just 2 wavelengths. Based on the previous work, we were able to identify just 2 wavelengths to build a very specific contrast function and make a very fast automated malaria detection instrument. This was the typical image we're able to get. And on the right part of the screen, you can see what we got when we design our contrast function. As you can see in the cells, when it's completely white, that means the cell is healthy. And when you have this black point, it shows that the cell is infected. And this black point is the byproduct of the parasite digestion in the food vacuole of the parasite within the blood cell. And we were able to show that this target is can be used to detect malaria. And the second image is also another contrast function we built from 2 wavelengths. And this gave led to another paper in a journal of spectral imaging. Just a few words about another aspect of this effort to fight against malaria. This is the prevention aspect. With the contribution of our partnership in Sweden, we built a remote spectroscopy instrument, which is able to detect an insect with the scale of a mosquito in a range of 1 kilometer. And using this instrument, as you can see, we have two parts. One of the telescope will send the light in the area of interest, and the second will collect the backscattering of the light when the insect cross the region of the interest. And because the insect has a frequency bit, we are able to get a footprint of the insect by using his frequency bit in the Fourier space. You have the sketch of the instrument here. We use a linear CCD. We have a receiver. Yes, we have a laser which will send the light, and the second hand is a receiver. And we have at the hand of the target a black support to reduce the backscattering and increase the sensitivity of the system. We have a number of papers in this area. The typical spectrum we collect is this one. This is our instrument from our lab. And what we are doing now is to combine this instrument with a weather station. And we are working on a project to do something like a mosquitoes forecast. The idea is to collect data during the whole year and be able to forecast the activities of the mosquitoes according to the season, according to the type of region, according to the type of field. So we are in progress on this work currently, and that is one of the efforts in the fight against malaria. This is some result we got with this instrument. When we collect the data, we can transform this data on this first spectra this image, let's say image, because it's a two-dimension data with time and a range. And the brighter part is the movement of the insect. In this case, it was a mosquito. So we are talking about an insect with a scale of a millimeter in the range of a kilometer detected. So on the technological aspect, this instrument is a very nice instrument. And yeah, you below you can see the typical data we can extract in the Fourier regime and collect the frequency of the insect. Here you have a representation taking into account the frequency as well as the time and the range. And you can see the event collected in this region. We also have three other type of instruments applied also in the medical area. The optical tweezer, we use this instrument to measure the elasticity, to differentiate the elasticity between the membrane of infected red blood cells and healthy red blood cells. And we have developed some methodologies to be able to propose a new target of malaria diagnosis using the mechanical elasticity of the membrane of the erythrocyte. This work was on the Buruli ulcer and is an application of diffuse reflectance. So the Buruli ulcer is a very spreaded sickness in the western part of Africa. And when it is earlier, the detection is earlier, it's possible to cure it. So the instrument we propose is something which is able to say in the very earlier state if we are facing to something which is going to lead to a Buruli ulcer or not. So it's mainly a USB spectrometer in the center part. The light, the backscattered light is collected using an optical fiber, as you can see in the green part. And around this, you have a number of LEDs with different wavelengths ranging from UV to near infrared region. And by playing with the different wavelengths, we are able to build a contrast function which is able to tell us if what we have on the skin is something which is going to lead to a Buruli ulcer. We have also built a light sheet imagery with the collaboration of our partner in the Lund University. This instrument is able to measure optical properties in a very dense area. The main idea is to modulate the light. Instead of using the classical technique we can see in the standard spectrophotometer, here we use the image. And the detection is done in this configuration in 90 degrees. So as I said, the main idea is to modulate the light, which is passing through the dense solution or dense material, with the idea that the ballistic photon will keep the memory of the modulation, and the diffuse part will lose the memory of we lose the memory of the modulation of the light. So by a post-processing technique, we are able to distinguish between the multiple scattered light, the multiple scattered photon, and the ballistic photon. So this instrument is used to make spectroscopy in the media, even if this media is dense. And we have a number of paper, and this instrument will be used for biological diagnosis. You know, when you collect a blood, you can make some diagnosis directly, use the blood collection. So that's one of the application of this instrument. And we have a patent on this instrument, and we have started a startup with this instrument in Sweden with a group of colleagues I'm working with. So where are we today? We exist as a lab, but as well as a network, let's say for 11 years now. We have trained a number of PhD students, a master's students as well. We have organized at least 5 workshops in different countries. We have constructed 5 instruments, and we, as I said, we are currently a PhD program in our network, and we are looking for partners working in the area of optical technologies, spectral images, image processing, chemometrics, and so on. And you are welcome to join us in this effort to help African scientists in the area of instrumentation. So as I said, this is the map of our interests, the application of the instrument we built in this network and in my lab as well. So thank you for your attention. Some picture of our collaborative activities. The first one in the left is the workshop in Nairobi in 2012. This is the workshop in Cape Coast in Ghana. This is one of the PhD who defended his PhD last year in Sweden. We have worked on this project in collaboration with Mikael. This is our very last workshop in Cape Coast. Yeah, this is some of our PhD students working on the construction of the instrument. So thank you very much for your attention. And now we can maybe through the question, maybe deepen some part of our talk. Thank you very much.